So today we're doing lipid biosynthesis, which we're going to look at the biosynthesis of fatty acids and eicosanoids. I don't know if I say that right. Assembly of fatty acids and glycerol into triacylglycerols and the biosynthesis of cholesterol. Even though we skip a lot of steps in that. But. So what do lipids do? Right, yeah, all of this stuff. Energy storage, they make up the membranes, anchors for membrane proteins, if you want to have proteins anchored into the membranes, cofactors for enzymes, signaling molecules, they make pigments, detergents, transporters, and antioxidants are lipids too, like vitamin E and so forth. Catabolism and anabolism of fatty acids proceeds via different pathways, right? Just like catabolism of sugar and anabolism of sugar are different, same thing with fats. And you can look at some similarities too. Catabolism of sugars produces acetyl-CoA, so same thing with fats. And it also produces reducing power in the form of NADPH for sugar, same thing for burning fats. And it takes place in the mitochondria. Well, glycolysis is in the cytosol, but once you finish with glycolysis, the rest is in the mitochondria. Anabolism of fats requires acetyl-CoA and malonyl-CoA. This is the rate-limiting reagent here is going to be your malonyl-CoA. Requires reducing power from NADPH, so you can, if you remember, anytime anabolic processes typically use NADPH, whereas catabolic processes typically produce NADH. And it takes place in the cytosol in animals, chloroplast, and in plants, where we're making these fats. So, and this just shows you the, different locations in the cells where we get NADPH production is in the cytosol via the pentose phosphate pathway and malic enzyme, etc. Isoprenoid and sterol synthesis in the cytosol, and fatty acid synthesis is in the cytosol also. No fatty acid oxidation in plant cells in the mitochondria. Where you do get fatty acid oxidation in the mitochondria in animal cells, acetyl-CoA production, ketone body synthesis, and fatty acid elongation occurs in the mitochondria. So, overview of fatty acid synthesis. Like I was saying, malonyl-CoA is the basically the activated form of acetate. So we start off with acetic acid, and we're going to add acetic acid 1 unit at a time to a growing chain of a fatty acid. And we do that by activating acetyl acid by converting it to malonyl-CoA. So do you guys know the structure of acetyl-CoA versus malonyl-CoA? I mean, acetyl-CoA is not up here, but can you guess what the difference is? Say again? Sort of, like, I mean, this, if you had a hydrogen here, CH3, that would be acetyl-CoA if we didn't have this. So what's the added group to acetate? What kind of group is this? Well, it's I mean, you could consider that a carbonyl, but having this extra oxygen makes it something else. What is that? That carbon's further oxidized. It's a not a methyl, but carboxylic acid. This is our carboxylic acid. Where do you think this will come from? Yeah, or bicarbonate. So this basically, malonyl-CoA is acetic acid or acetyl-CoA that's been carboxylated, okay? And it's basically a way to activate acetyl-CoA so you can get condensation with another acetyl-CoA.
So acetyl-CoA is that last.
Yeah, everything here over would just be Acetyl-CoA. The only difference is that rather than having another hydrogen bound to this carbon, you have a carboxylic acid. So you've replaced the hydrogen with a carboxylic acid or carbon dioxide, right? So like I was getting at, malonyl-CoA is formed by acetyl-CoA and bicarbonate, or carbon dioxide. And it's catalyzed by the enzyme acetyl-CoA carboxylase. So acetyl-CoA carboxylase is the entry step in the fatty acid synthesis. Has 3 units, and we've already talked about this many times. The coenzyme that you always see when you see bicarbonate and ATP, when you're shifting a carboxylic acid group from one to the other, carboxylating things, is what? Right, biotin. You remember that? It's on a tether. It picks up the carbon, dioxide on one side and delivers it to its substrate on the other end of the enzyme. So in animals, all 3 of the subunits are on one polypeptide chain. And bicarbonate is the source for the carbon dioxide to make the malonyl-CoA. It's a 2-step reaction that's similar to carboxylations catalyzed by pyruvate carboxylase and propionyl-CoA carboxylase. You guys remember pyruvate carboxylase? That gluconeogenesis. Anyways, carbon dioxide binds to the biotin. It's activated by the attachment to the nitrogen in the ring of the biotin, if you remember. And the reaction with ATP produces that carbamyl phosphate. So let's see this. So if you remember, biotin has this little ring structure, and it's on it's covalently bound to an enzyme based on an amide bond with the end of a lysine chain forms an amide with the carboxylic acid of the biotin itself. So it's on this long hydrocarbon chain, and it's able to go from one subunit to the other. So it goes from the biotin carboxylase, picks up the carbon dioxide here, then it'll transfer to the other subunit, the trans-carboxylase, where it adds that carboxyl group to acetyl-CoA, and you get the formation of malonyl-CoA. So, and catalyze. So the synthesis of fatty acid is catalyzed by fatty acid synthase. So here we have to have malonyl-CoA before we can synthesize fatty acids, and then the synthesis of fatty acids is catalyzed by fatty acid synthase. So can you remember that name of that enzyme? Fatty acid synthase is going to do what? Synthesize fatty acids. That's an enzyme that's named well, right? So it catalyzes a repeating 4-step sequence that elongates the fatty acyl chain by 2 carbons at each step. So I guess this is figure 21.2. We use NADPH as an electron donor, and it uses 2 enzyme-bound thiol groups as activating groups. Fatty acid synthase 1 is what we have. It's found in vertebrates and fungi, and then there's fatty acid synthase 2 in plants and bacteria. And there's 4 steps, and it's nearly the reverse of the beta oxidation steps, right? 4 steps, you have malonyl group and an acetyl group. You get condensation, decarboxylation. So then you have, alpha beta keto group. That gets reduced from NADPH, and it turns it into an alcohol. Then with the alcohol, we don't need to change oxidation state. We just remove the water, and you have an ene. From the dehydration group, you form an ene double bond here. And then you reduce that double bond again, requiring another NADPH, and you have a saturated acyl that's 2 carbons longer than what you started with. And you'll repeat that over and over again until you get to 16 carbons long. So So fatty acid synthase 1 versus fatty acid synthase 2. Fatty acid synthase 1 is a single polypeptide chain in vertebrates, leads to a single product, palmitate, 16, a fatty acid that's 16 carbons long. C15 and C16 are formed from the acetyl-CoA used to prime the reaction. So those first two, at the end, they're the first ones. Wind up being from the initial acetyl-CoA. Fatty acid synthase 2 is made from separate diffusible enzymes. So these are all one chain. These are separate diffusible enzymes in plants and bacteria. Makes many products, saturated, unsaturated, branched, many lengths. And we find that in plants and bacteria. This is an image of fatty acid synthase type 1. And we got our four different subunits there where we get all the reactions taking place. So fatty acid synthesis, the overall goal is to attach 2 acetate units from malonyl-CoA to a growing chain and then reduce it. Reactions involve the 4 cycles. It's basically the reverse of beta oxidation, right? So we have condensation, and that produces a beta keto acid. Reduction of carbonyl to a hydroxyl, so you take that ketone group and convert it to an alcoholic group. Remove the water from that, which causes a trans-alkene group, and then you reduce that alkene to an alkane, and we have our saturated fatty acid. That's 2 carbons longer. The growing chain's initially attached to the enzyme via a thioester linkage, and during condensation, the growing chain is transferred to the acyl carrier protein. I think we talked about the acyl carrier protein briefly before. Do you guys remember that? Remember coenzyme A and this acyl carrier protein share the same, they share the same functional groups, right? It's basically, acyl carrier protein has the same active group that carries the fatty acids or the growing fatty acids that acetyl-CoA has, but acetyl-CoA is diffusible and can go from one enzyme to the other. It just carries fatty acids around the cell, as in coenzyme A conjugated lipid. But acyl carrier protein is specific for fatty acid synthesis. So rather than using acetyl-CoA, it's a large protein and it all stays with the fatty acid synthase. After the second reduction step, the elongated chain is transferred back to the fatty acid synthase. So malonyl-CoA and acetyl-CoA, or a longer fatty acid chain, depending on where we are in the process, are bound to fatty acid synthase 1, and they're bound via a thioester terminus or a cysteine of the fatty acid synthase. And that, thioester, it's a higher energy bond, it activates that acyl group. Step 2 is you have a condensation reaction that attaches 2 carbons from the malonyl-CoA to the attached acetyl-CoA or longer fatty acid chain. And during this process, we decarboxylate the malonyl-CoA. So we lose 1 carbon as carbon dioxide, and then, so you lose 1 carbon. Those electrons that were attached to that carbon dioxide before have to go somewhere, and they attack the other 2 carbon fatty acid, okay? So this decarboxylation facilitates the reaction. We have to activate the acetyl-CoA first before we can form the malonyl, and that energy stored in activation of it, adding the carboxylic acid, is the energy that drives this condensation forward. And it creates that beta keto intermediate. So the first step, you have your malonyl-CoA. You see the electrons drop down, which basically forms carbon dioxide here. These electrons have to go somewhere, and they attack that the carbonyl on the acetyl-CoA that's bound to the fatty acid synthase. And then you end up with 2 carbons longer with a beta keto group on the growing fatty acid chain. So we're ready for step 2 then, which is going to be, what are we going to do? This is fairly oxidized. We're trying to make a reduced fuel. So we need to reduce this carbonyl, right? And what are we going to use to reduce the carbonyl? Where do we get the reducing power from?
NADPH?
Yes, NADPH is where we get the electrons and reducing power to reduce that carbonyl. So step 2 is the reduction. NADPH reduces the beta-keto intermediate. Then we go on to step 3. Converted that beta-keto to an alcohol, a beta alcohol. We remove water, which forms a transalkene, double bond there. And then we reduce that double bond, and we have the second reduction, which requires, again, NADPH. And that yields a saturated alkene. And here they show you all those steps again. Reduction, go from the beta-keto to the beta-alcohol. Then step 3, we remove water. See this H2O, and that forms the alkene. Double bond there. That requires reduction. Reducing power again comes from NADPH, and we have the saturated acyl group that's lengthened by 2 carbons. And to make palmitate, you just do this over and over and over again till we get, 16 carbons long.
What's magic about 16? Why doesn't it keep going?
You can keep going, but the enzyme won't bind to these anymore. So you can have longer fatty acids, but palmitate is just the limit of this, the fatty acyl synthase 1. So I don't know if there's something magic about it.
I don't mean magic. I don't see what, from the end that it's acting on, it looks the same.
Yeah, but as you add more and more carbons, it's going to be less and less, it'll be more and more hydrophobic, basically. So you'll need an enzyme with maybe a larger hydrophobic pocket to support that long hydrophobic tail as you keep getting longer and longer. So this is the overall process. Just adds more and more. So like we had discussed before, the acyl carrier protein basically serves as the shuttle in the fatty acid synthase. And it's the same active group that we see in coenzyme A, but rather than it being bound to AMP, it's bound to a protein, a large protein. So delivers the acetate in the first step, or the malonate, and all the next steps to the fatty acid synthase. Shuttles the growing chain from one active site to another during that 4-step reaction. So we only see this in the fatty acid synthesis. Remember, from beta oxidation, it was Acetyl-CoA that delivered that and carried the fatty acids. So charging this acyl carrier protein and fatty acid synthase 1 with an acyl group activates them. You have 2 thiol groups. Must be charged with the correct acyl groups before you can get condensation reaction to begin. The thiol from the 4-phosphopantethine in the ACP, and this is the phosphopantethine right here, this whole group. And the thiol from the cysteine in the fatty acid synthase. I think they showed you this before. You have this thiol here. That's part of the fatty acid synthase that where you get that fatty acid. So both of those have to be charged first. And then the acetyl group from acetyl-CoA is then transferred to the acetyl carrier protein, catalyzed by malonyl acetyl-CoA transferase. The acyl carrier protein passes this acetate to the cysteine of the beta keto acyl carrier protein synthase domain of fatty acid synthase 1. And the acyl carrier protein thiol group is then recharged with malonyl from malonyl-CoA. So this is the charging activation with acyl carrier protein in the 4-step sequence of mammalian fatty acid synthase. So we have, start off this, you have the fatty acid synthase, the acyl carrier protein with nothing on its pantethine group, and then acetyl-coenzyme A will come in, transfer that acetate group to the thiol on the fatty acid synthase, and then malonyl-CoA will come in and transfer its malonyl group to the thiol of the pantethine acyl carrier protein group. And then you can get condensation, decarboxylation here, yields the condensation reaction, and then the beta keto acid is then bound to that acyl carrier protein, and it'll go from one subunit to the other, one active site, not subunit, but one active site to the other on the acyl carrier protein for the rest of the, the process. And then you end up back on this thiol group, 2 carbons longer, like here. So this is charged already. Then you get another malonyl-CoA that comes in and adds on to that acyl carrier protein. So then the cycle will continue, and it'll end up being another 2 carbons longer. Goes from the acyl carrier protein to the thiol group on the fatty acid synthase. Another malonyl-CoA comes in and transfers its malonyl to the acyl carrier protein, and it goes over and over and over again. And these are what I just described, but with bigger pictures. So we have the malonyl acetyl-CoA ACP transferase. Acetyl-CoA comes in. The acetate unit gets transferred from the coenzyme A to the acyl carrier protein. Or no, I mean not the acyl carrier protein, the fatty acid synthase in the beta keto ACP synthase subunit. And then the malonyl-CoA will come in and get transferred to the acyl carrier protein. Decarboxylation occurs, and you get the formation of the beta keto acid that is now, attached to the tail end of that acetate that was the malonyl-CoA that's still on the acyl carrier protein. Then we get reduction to an alcohol. Then we get dehydration to that ene group, the trans-delta butanoyl ACP, and it won't always be butanoyl. It depends on how many carbons are being added, or how many carbons it was added to. And then reduction again from that to the saturated fatty acid. That's 2 carbons longer. And then that saturated fatty acid has to be transferred from the acyl carrier protein to the thiol on the fatty acid synthase. And then we get the acyl carrier protein gets recharged with another malonyl-CoA group, and the whole process will go again. So again, fatty acid synthesis in detail. Step 1, condensation, and it's basically a Claisian condensation reaction catalyzed by beta keto-acyl-ACP synthase. Coupling the condensation to decarboxylation of malonyl-CoA makes this reaction energetically favorable. Carbonyl at the C3 is reduced to the D-beta hydroxybutyryl ACP. NADPH, again, is the electron donor, and that's catalyzed by beta-keto-acyl-ACP reductase. Then the hydroxy group and a hydrogen is removed from the C2 and C3 of the beta-hydroxybutyryl ACP to form the trans-delta butaenoyl, the ene group, catalyzed by beta-hydroxy-ACP dehydratase. Dehydratase removing water. Fatty acid synthesis in detail, step 4. Then that ene group needs reduction. And that we get those from that reducing power from NADPH, and that ene group's reduced to the saturated ACP, catalyzed by enoyl ACP reductase. And these are the enzymes in order, and then we get the chain transfer and charging again, following that order. So the transferase and fatty acid synthase reactions are repeated in new rounds. Product of the first round is the butyryl ACP bound to the phosphopantethene of that group on the ACP. The butyryl group is then transferred to the cysteine of the beta-keto-acyl ACP synthase. In the first round, Acetyl-CoA was bound there. The new malonyl-CoA binds the ACP, and after the new round, a 4-step 6-carbon product is made bound to the ACP. And this shows you the beginning of the second round. So we have the butyryl group bound to the fatty acid synthase. Another malonyl comes in. Malonyl-CoA, that malonyl gets transferred to the ACP, and we get condensation again. It's 2 carbons longer. And we go through, after condensation, there's reduction, dehydration, and then reduction again, right? So the keto becomes an alcohol, the alcohol an ene, and then the ene becomes saturated after it's reduced again. And then the stoichiometry of the synthesis of palmitate. Like I said, I don't like you memorizing like numbers and things like that. But take 7 acetyl-CoAs that are carboxylated to make 7 malonyl-CoAs using ATP. So each one of those 7 carbon dioxide, 7 ATPs, we get 7 malonyl-CoAs. 7 cycles of condensation, reduction, dehydration, and reduction again using NADPH to reduce the beta-keto and the trans double bond. We end up with 1 palmitate, 7 carbon dioxides from before just released again, 8 coenzymes A's from here, 14 NADPHs are used, so we end up with 14. So you remember we got to reduce each one of these malonyls twice, right? First we form the beta-keto, dehydrate it to the ene, and it requires another NADH, so 2 NADs for each malonyl. So acetyl-CoA is transported into the cytosol for fatty acid synthesis. In non-photosynthetic eukaryotes this happens. So acetyl-CoA is made in the mitochondria, right? During the, either from fatty acid catabolism or from pyruvate, right? We get acetyl-CoA made in the mitochondria that would be used for the TCA cycle, but if you have a high energetic state of the cell, that acetyl-CoA would be better stored as energy rather than burned, right? So acetyl-CoA is made in the mitochondria, but fatty acids are made in the cytosol, so this acetyl-CoA has to be transported into the cytosol, and that costs energy to transport that across the mitochondrial membrane. So the cost of fatty acid synthesis is 3 ATPs per 2 carbon unit. So 2 of them comes from, transport. Where does this other one come from? Do you guys remember? That first step Acetyl-CoA carboxylase requires an ATP to activate that carboxyl group, so it'll be added, you know, to the acetyl group, the acetate group on acetyl-CoA. So, fatty acid synthesis occurs in cell compartments where NADPH levels are high. Cytosol for animals and yeast. We get high NADPH in the chloroplast in plants, right, from the light reactions. Sources of NADPH in adipocytes are the pentose phosphate pathway and malic enzyme. NADPH as malate converts to pyruvate and carbon dioxide. And hepatocytes in the mammary gland pentose phosphate pathways where we get our NADPH. NADPH made as glucose 6-phosphate converts to ribulose 6-phosphate. And then in plants, photosynthesis drives NADPH production. This is the same figure from before, isn't it? And this shows you the pathways for NADPH production. So malic enzyme, malate gets oxidized to pyruvate, and in the process we produce NADPH. Like with that, glucose 6-phosphate via the pentose phosphate pathway will produce 2 NADPHs, and then also ribulose 5-phosphate, which provides a 5-carbon sugar for nucleic acids. Acetyl-CoA is generated in the mitochondria and shuttled to the cytosol as citrate. Most eukaryotes, acetyl-CoA for lipid synthesis is made in the mitochondria, but the lipid synthesis occurs in the cytosol. There's no way for acetyl-CoA to cross that mitochondrial inner membrane to the cytosol, so the acetyl-CoA is converted to citrate. Do you guys remember how that works? Acetyl-CoA plus oxaloacetate forms citrate. Same reaction in the citric acid cycle or the TCA cycle. So it's that first step in the TCA cycle. We condense acetyl-CoA with oxaloacetate that produces citrate. Remember, citrate is one of the regulators of glycolysis. Citrate levels are really high. Do you remember what enzyme it acts as a allosteric inhibitor for? Phosphofructokinase 1, the entry step into glycolysis. So if citrate levels are building up in the TCA cycle, they're going to start to be transported out into the cytosol, which will slow down glycolysis and also provide acetate units for the production of fatty acids, right? So citrate then, it passes through a citrate transporter. So that's how the acetyl-CoA can leave the mitochondria. Citrate now in the cytosol is cleaved by citrate lyase that regenerates acetyl-CoA and oxaloacetate and requires ATP for that reaction. Acetyl-CoA can now be used for the lipid synthesis. And what happens to the oxaloacetate? Right, because there's no oxaloacetate transporter either. Do you guys remember that? So that malate dehydrogenase in the cytosol reduces oxaloacetate to malate. Do you guys remember the malate oxaloacetate shuttle? 2 potential fates of malate can be converted to NADPH in the cytosol and pyruvate via the malic enzyme. So that's one of the sources of NADPH used for lipid synthesis. And then the pyruvate in the cytosol sent back to the mitochondria via the pyruvate transporter and converted back into oxaloacetate in the mitochondria by pyruvate carboxylase. And that requires ATP to convert it back to oxaloacetate. Can be transported back to the mitochondria via the malate alpha-ketoglutarate transporter. Malate is reoxidized into oxaloacetate. And this figure shows you the 2 possible mechanisms. So we have the matrix in the mitochondria and then the cytosol. So acetyl-CoA can come from multiple sources. It gets citrate synthase will convert acetyl-CoA and the oxaloacetate to citrate. It can be transported out into the cytosol. The citrate can be stripped of that acetyl-CoA and we produce oxaloacetate again. That requires ATP. Then oxaloacetate can be reduced to malate and transported back, or Malic enzyme can be further oxidized, and you regenerate that NADPH, which you'll need, right, for fatty acid synthesis. And that produces from malate goes to pyruvate. Pyruvate can then be transported in, or pyruvate carboxylate converts it back to oxaloacetate. And that oxaloacetate can then serve, again, as a precursor to citrate and continue that TCA cycle. Or it's a precursor to amino acid synthesis. So fatty acid synthesis is tightly regulated via the Acetyl-CoA carboxylase. So this is the regulated step, because this is the entry step for that acetyl-CoA to enter into fatty acid synthesis. Acetyl-CoA is feedback inhibited by palmitate-CoA, so that ultimate product that you're making with the fatty acid synthase is palmitate-CoA. When you increase the levels of this, it'll inhibit acetyl-CoA from entering that pathway, right? And acetyl-CoA carboxylase is activated by citrate, right? So citrate, again, if the TCA cycle's backed up, we've got plenty of energy, plenty of citrate there. You've sent too much acetyl-CoA into the TCA cycle, you form citrate. Concentrations of citrate are high, they get transported to the cytosol. That citrate in the cytosol will activate acetyl-CoA carboxylase, thus activating fatty acid synthesis. And so the acetyl-CoA that was used to make citrate can then be used to store that energy again as fat. Citrate signals excess energy to be converted to fat. When acetyl-CoA in the mitochondria is high, it's converted to citrate. Citrate's exported to the cytosol again. So importance of citrate to the regulation of fatty acid synthase. So in animals, citrate stimulates fatty acid synthesis. Like I said before, it's the precursor for acetyl-CoA, sent to the cytosol and cleaved to become acetyl-CoA when acetyl-CoA and ATP is high, right? Allosteric activator of acetyl-CoA carboxylase, and it's an inhibitor of phosphofructokinase 1. So if you have citrate in the cytosol, it's going to inhibit burning sugar, and it's going to activate storing acetate as fat. So it's a good regulator of energy. Reduces glycolysis, it increases fatty acid synthesis. So acetyl-CoA carboxylase is regulated by covalent modification. It's inhibited when energy is needed, right? So we don't want to store fat when we need energy. So glucagon and epinephrine, if you remember, are the hormones that tell us that we need more energy and we don't want to store energy. Insulin is the hormone that tells us to store the energy, right? You guys remember that? So glucagon and epinephrine reduce sensitivity of the citrate activation. It leads to the phosphorylation and inactivation of acetyl-CoA carboxylase. Acetyl-CoA carboxylase is active as a dephosphorylated monomer. When the dephosphorylated ACC polymerizes into a long inactive filament. So you can have acetyl-CoA carboxylase. It's active as a monomer, but as it polymerizes, it's inactive. So it's dephosphorylated, it polymerizes, and it's inactive. When it's active, long inactive filaments. ACC is active as dephosphorylated. Phosphorylation reverses the Wait, does this make sense here? Or did I jump the gun on my reading of this slide? So glucagon and epinephrine will reduce the sensitivity of acetyl-CoA carboxylase to citrate activation. Leads to the phosphorylation and inactivation of acetyl-CoA carboxylase. So acetyl-CoA is active as dephosphorylated monomers, but when dephosphorylated, it'll polymerize in long inactive filaments. So it prefers to be phosphorylated in monomers. Phosphorylation reverses that polymerization process. Here they show you like a phosphorylated form of acetyl-CoA carboxylase. I mean, not a phosphorylated, a polymerized form of acetyl-CoA carboxylase. So acetyl-CoA carboxylase is blocked by glucagon epinephrine, triggers phosphorylation inactivation, and then palmitate-CoA will also block that. Whereas acetyl-CoA is activated by citrate. Let me see. So I like this other slide better than this one, just to clarify that regulation. So phosphorylated protomer of the acetyl-CoA carboxylase is inactive. Citrate activates it, causes the dephosphorylated polymer acetyl-CoA carboxylase to be active. Whereas palmitate-CoA shifts it to the inactive form. So what did that, our other PowerPoint just say? Yeah, it leads to the phosphorylation and inactivation when dephosphorylated polymer. So it's kind of the opposite what the other slide is saying. So this is a mistake here. So phosphorylated protomer is inactive. Dephosphorylated polymer is active, and you get an antagonistic effect of insulin produced when the blood glucose is high, is attributed to the activation of protein phosphatase. Right? So insulin, you want to store energy, activates protein phosphatase, removes the phosphate groups from acetyl-CoA carboxylase, which causes the polymer, the dephosphorylated polymer acetyl-CoA carboxylase to be in the active form, so it's going to store acetyl-CoA as fats. So local metabolites also, we already talked about palmitate-CoA, right? The product promotes the inactive conformation, diminishing the product of production of malonyl-CoA, the precursor of fatty acid synthesis, so it's feedback inhibition. So are you guys more clear or completely confused? This one's a lot better than, yeah. So I'll try to switch it up and go back to these figures, okay? And here's another figure here from the old book, which doesn't show the polymerization. So kinases phosphorylate it. High citrate activates low citrate. Low fatty Acyl-CoA inhibits. Dephosphorylated form, low citrate will activate. High fatty Acyl-CoA inhibits. So I don't like that figure either as much as this little animation is better, I think. So regulation of fatty acid synthesis in plants and bacteria does not rely on citrate or phosphorylation. The plant ACC is activated by high magnesium and pH, and bacteria use lipids for membranes, not energy storage. So have complex regulation with guanine nucleotides, and that coordinates with cell growth. So if you have increased in cell growth, you'll have to increase the amount of lipids that are being made so you can divide. Changes in gene expression. So the fatty acids and eicosanoids bind transcription factors called peroxisome proliferator activated receptors, PPARs, and reciprocal regulation. Malonyl-CoA inhibits fatty acid import into the mitochondria. One of many ways to ensure that fat synthesized and oxidation doesn't occur simultaneously. If they were occurring simultaneously, what kind of cycle would that be called? A futile cycle, right? And you remember, you're burning all that ATP to make it, you're getting less ATP back out when you burn it. So it's kind of just energy costing. I guess if you want to burn fat, it might be good, right? Make a drug that dysregulates fatty acid synthesis and fatty acid burning. Be a good way to a fat burning drug, right? As long as what?
So long as your cells don't starve from it.
Yes. So elongation systems in the endoplasmic reticulum and mitochondria can create longer fatty acids. So you're asking, why does it stop at 16? Just fatty acid synthase 1 stops at 16, but then you can add more 2 carbon units to it. To stearate is the most common process, so you just add another 2 carbon unit. And that happens in the endoplasmic reticulum and the mitochondria, not in the cytosol. So palmitate can then be desaturated also. So we typically just produce the saturated, but then we also need to desaturate. So palmitate can be converted to palmitoylate delta 9, which has 1 double bond, 1 ene group. Stearate can be converted to oleate, which is another 1 ene group, and that delta 9 is. Do you know what the delta 9 is? It's the 9th carbon down. Is where you get the ene group. And you know what the omega stands for? You've heard of omega 3 and omega 6? That's up from the back of the fatty acid. So also known as fatty acid desaturases. Does make sense as an enzyme name or a class of enzymes. They're desaturases. They remove the saturation. Requires NADPH. Enzyme uses cytochrome B5 and cytochrome B5 reductase. And it's a delta 9 desaturase. It reduces the bond that's between C9 and C10. Vertebrate fatty acyl desaturase is non-heme iron-containing mixed function oxidase. Oxygen accepts 4 electrons from 2 substrates. 2 electrons come from the saturated fatty acid, and then 2 electrons will come from a ferrous state of cytochrome. So we reduce the oxygen to water, and then 2 electrons come from the fatty acid, 2 come from the cytochrome B5. And then for that enzyme to be regenerated, that cytochrome B5 is going to require electrons to come from NADPH. So we're using an NADPH to actually oxidize a fatty acid. Does that make sense? Doesn't make sense so much, but it does because we have to regenerate the iron here in the desaturation enzyme. So we take one oxygen, two hydrogen ions, a saturated fatty Acetyl-CoA, its electrons and the electrons from the cytochrome B5 transfer to the oxygen to form water, and it's desaturated at one bond, delta 9. And then the cytochrome B5 has to be re-reduced. It gets the electrons from FADH2 from cytochrome B5 reductase, which gets its electrons from NADPH.
Where was it that we generated the NADPH again?
So, multiple places, right? Malic enzyme can generate it. The pentose phosphate pathway can generate NADPH, among others, right? But those were the two big ones. So plants can desaturate beyond the C9. So humans have delta 4, delta 5, delta 6, and delta 9 desaturations, but we can't desaturate beyond, that ninth, delta 9. Plants, they'll produce lienolate, which is 18,2, delta 9, and delta 12. Alpha lienolate, which is delta 9, delta 12, delta 15. And these fatty acids are considered essential because we can't make them ourselves, so we have to acquire them in our diet. Polyunsaturated fatty acids help control membrane fluidity, and they're the precursors to eicosanoids. So we have to have those eicosanoids. So, unlike mammal desaturases, plant desaturases do not oxidize free fatty acids. They oxidize fatty acids that are bound to glycerol, such as like phospholipids. So phospholipids, they can just oxidize the fatty acid right there in the phospholipid. The free desaturated fatty acids can then be hydrolyzed to form glycerol backbone. So here you have phosphatidylcholine, which is a phosphoglyceride. Desaturase can, desaturate that multiple times, and then this can be cleaved out of that phospholipid. Oxidases, monooxygenases, and dioxygenases. Many enzymes use oxygen as the electron acceptor, but not all of them incorporate oxygen into the product. So oxidases do not incorporate oxygen into the product. Oxygen atoms usually end up as H2O2. Oxygenases do not incorporate oxygen into the product. Monooxygenases incorporate one oxygen atom into the product, and dioxygenases incorporate both oxygens. So oxygenases do, oxidases do not. Does that make sense? So monooxygenases incorporate one oxygen into its product. You have AH plus BH2 plus oxygen, and you've converted that AH to an alcohol. You've incorporated one oxygen, the other oxygen got reduced to water. Product is often hydroxylated, so it's a lot of hydroxylases are mixed function oxygenases. Examples are phenylalanine hydroxylase, hydroxylates phenylalanine, and that forms tyrosine. And then you have tyrosine hydroxylase, goes even further, right? Adds another hydroxyl group. So cytochrome P450s are monooxygenases. So they're important in drug metabolism. The majority of our drugs are metabolized by cytochrome P450s. They hydroxylate nonpolar molecules. So these mixed function oxidases, or the cytochrome monooxygenases, they're there in our liver to clear out what's known as xenobiotics. Have you heard of those? They're like foreign biologically active molecules. So often they're hydrocarbons. Like if we cook our food on the barbecue, we'll get a lot of hydrocarbons. These cytochrome P450s, in order to get rid of them from your body, it wants to make them more water soluble, so you can just excrete them in your in your urine, basically, right? So you add hydroxyl groups, and these cytochrome monooxygenases add hydroxyl groups to it using oxygen to make them more water soluble. If two drugs or alcohol and a drug use the same enzyme, they'll compete and the levels of the drug or alcohol will not be cleared as quickly. So that's where you get these drug interactions. They're competing for the same metabolizing enzyme. And so if you're taking it on a regular basis and you happen to take another drug that competes for the same metabolizing enzyme, you'll boost the levels in your blood, and if you're not careful, that you can boost them to a level that's toxic, right? So it can be deadly, right? Dioxygenases incorporate two oxygens into the product, and they're usually metalloproteins. The active site have an iron or manganese. Reactions often involve opening of an aromatic ring, and they use to convert catechols to ketones. Examples are catechol 1,2 dioxygenase or tryptophan 2,3 dioxygenase. So here we have tryptophan 2,3 dioxygenase. We have the indole ring here. Oxygen comes in and it splits that indole ring, and you form N-formyl ketonurine. I don't know how you say that. But you've added oxygen here and oxygen here in the form of carbonyls, right? Eicosanoids are potent short-range hormones made from acidic acid or acidic. And eicosanoid hormones include prostaglandins, leukotrienes, and thromboxans. They're created from the acridonic 20, it's got 4 double bonds there. I don't know if you've seen the pictures with it bent around one of the non-double bonds. It kind of forms like a structure like this. And in response to stimuli, phospholipase A2 is activated and it attacks the C2 fatty acid, releasing acridonate. And then that can be used to make prostaglandins like this. So this is acridonic acid. Got the 4 double bonds. Cyclooxygenase activity of COX adds a peroxide group here and here, and then it also cyclizes that ring structure there, right? And then the peroxidase activity will then occurs, and you get the reduction of that peroxide there. And then you have prostaglandins and thromboxans can be made from that. So, and this is where we come into COX-1 and COX-2. Have you heard of COX-1 or COX-2 inhibitors? Or just COX inhibitors in general? They have some up here. This is where aspirin and ibuprofen function, where you get the analgesia. We block the formation of these prostaglandins and thromboxans, right? COX-1 catalyzes the synthesis of prostaglandins, and that regulates gastric mucin secretion. So you've always heard of the side effects. People taking too much aspirin or ibuprofen is hard on your stomach, right? That's because COX-1 regulates the prostaglandins that regulate gastric mucin secretion. So that's a side effect that we don't want, right, of COX-1 or COX inhibitors. If we have a general COX inhibitors with ibuprofen and aspirin R, they'll block both of these. This is the side effect, and this is the activity we're looking for in the drug, right? COX-2 inhibition catalyzes the synthesis of prostaglandins that mediate pain, inflammation, and fever. So this is what we're looking for. Aspirin, ibuprofen, acetaminophen inhibit COX-2, but they're not specific to COX-2. But there are some COX-2 specific NSAIDs. Do you guys remember those, or have you heard of those? No? They pulled them off the market, or remember? Celebrex, I think, maybe Vioxx, I can't remember. But they were specific to COX-2, but they had this, do you guys remember what the bad side effects were that they produced? Increased rate of heart malfunction and deaths from heart disease, okay? So, but they're for people who have like chronic arthritis who need to take something every day for that inflammation. And if you're taking aspirin and ibuprofen every day, that causes people to have stomach problems, right?
Stomach doesn't make the mucus that protects it from the stomach acid.
Right.
So it just gets through.
Yeah. Makes it uncomfortable. So NSAIDs inhibit cyclooxygenase activity. Aspirin, which is acetylsalicylate, is an irreversible inhibitor. So irreversible inhibitors typically have covalent attachments, right? And what it does is aspirin is acetylsalicylic acid. This acetyl group gets transferred to serines in the active site, and it blocks the active site in both COX isozymes. So it basically acetylates things. It also has activity all over the body from this acetylation, right? It blocks what? Blood clotting, things like that. That's why they say you should eat a little bit of aspirin every day, or they did. I don't know if they still say that. Because it's good for your heart, supposedly. Ibuprofen and naproxen are competitive inhibitors, and they resemble the substrate, but they also block the active site in both enzymes. So neither of these are specific for COX-1 or COX-2. And this shows you their structure. So aspirin, this acetate group, acetylates the serines in the active site, and you're left with salicylic acid as its product. And then that acetylated is inactive because you've blocked that active site from activity. And then ibuprofen and naproxen are just competitive, non-irreversible, or they are reversible inhibitors. COX-2, oh, I was right, Vioxx and Celebrex. So COX-2 specific inhibitors have a checkered history, like I just said, right? So they were developed to inhibit prostaglandin formation without harming the stomach. Includes Vioxx, Bextra, and Celebrex. Vioxx and Bextra are removed from the market due to the increased rates of stroke and heart attack. May disrupt the balance between blood thinning, prostacyclin, and blood clotting thromboxans, I suppose. So leukotriene synthesis also begins with arachidonic acid. So oxygen is added to the arachidonate via lipoxygenase, and then creates a species that differ in the position of the OOH group. So this is the leukotriene synthesis. So it's just different leukotrienes, different where this OOH gets added. So fat, right, if you guys remember, are triacylglycerols and phospholipids. And we store fat for fuel. Plants store it in seeds and nuts. Typically humans has about 15 kilograms of fat if you're about 70 kilograms large, enough to last us 12 weeks, compared with 12 hours worth of glycogen in the liver. If we didn't have any fat at all, we wouldn't last unless we, half a day for starving, right? Whereas fat keep us going, until we can get rescued on that deserted island.
Does that mean so long as you have the vitamins that you require, you just not eat for 12 weeks, the average person?
Yeah, but you wouldn't want to do that. And then animals and plants and bacteria make phospholipids for the cell membrane. So we're going to talk about this synthesis. The precursor for the backbone of fat and phospholipids is glycerol 3-phosphate, right? Because they're glycerophospholipids or triglycerides. for the storage. Most glycerol 3-phosphate comes from siphoning off dihydroxyacetone phosphate. And where do we get dihydroxyacetone phosphate from in animals? Yes, glycolysis. Does it say, oh. See, I asked the obvious questions in class. So, and that siphons is off via glycerol 3-phosphate dehydrogenase. So this enzyme, we're making glycerol dehydrogenase from dihydroxyacetone phosphate, so the enzyme's kind of named in the reverse direction, right? Because we could also take glycerol 3-phosphate, the enzyme would catalyze it the opposite direction too. Some glycerol 3-phosphate's made from glycerol via glycerol kinase. It's the minor pathway in the liver and kidney. And then acyltransferases will attach 2 fatty acids to glycerol 3-phosphate. So that makes sense. You're taking acyl groups, transferring them to glycerol. Acyltransferase. Phosphatidic acid is the precursor to triacylglycerols and phospholipids, made from glycerol 3-phosphate and 2 fatty acids. Fatty acids are attached via acyltransferase, and it releases coenzyme A, because all the acyl groups they're not free in the cell, they're always carried around by coenzyme A, right? Advantage of making phosphatidic acid, it can then be made into a triacylglycerol or a phospholipid. So once you make the phosphatidic acid, which is just a diacylglycerol with a phosphate added to that glycerol, is what phosphatidic acid is. I think they show you the picture here, right? And this way we got 2 acyl groups, 2 fatty acids, and then the glycerol with a phosphate, so it's kind of an activated group. This will be used either for making a triacylglycerol or for making a membrane lipid, right? Because already this is kind of a phospholipid here, right? And yeah, the reactions for doing this are pretty simple. We have the fatty acids, these are high energy, they're kind of activated in this state with the thiol esters, and they just transfer directly to the alcohol groups on the glycerol, releasing the coenzyme. So to make triacylglycerols, phosphatidic acid is dephosphorylated and then acetylated, or acylated, not acetylated. Phosphatidic acid phosphatase, again, phosphatases do what? Remove phosphate groups, right? Protein phosphatases remove phosphate groups from proteins for regulation. Phosphatidic acid phosphatase removes the phosphate group from the glycerol. Removes the 3-phosphate from the phosphatidic acid, yields 1,2-diacylglycerol. The third carbon is then acylated. I keep wanting to say acetylated. Acylated with the third fatty acid, and it yields a triacylglycerol. That's what we see here. So phosphatidic acid phosphatase. You have acyltransferase then. This is an activated acyl group. Can then be added to that free alcohol group, and you have a triacylglycerol, which is then used just to store those triacyl, or those acyl groups. Regulation of triacylglycerol synthesis, again, is by insulin, right? Insulin tells us to store energy. So insulin is going to, result in the stimulation of triacylglycerol synthesis. Lack of insulin results in increased lipolysis, increased fatty acid oxidation, and ketone generation, right? If you don't have insulin, you're not going to be able to take up sugar. Cells need energy, so as an alternative to sugar, they burn fat in the form of ketones. So that's where you get the diabetic ketoacidosis, right? You've heard of that? So sometimes to ketones in the citric acid cycle intermediates, they react with acetyl-CoA and are depleted. Failure to synthesize fatty acids. So this is, if you don't have, when don't you have insulin in diabetes? It's that
You mean which type?
yeah. It's that type 1,
Type 1.
right? So that's that.
I don't do this to myself type.
You what? Yeah. And if you ever seen people with type 1 diabetes, they're typically pretty skinny, right? Versus the other type when you're older. Huh?
I should say that was a generalization.
Yeah. So regulation of triacylglycerol synthesis of insulin. Increase insulin. Typically we secrete insulin when we have glucose. It's going to activate glucose conversion to acetyl-CoA and amino acid conversion to acetyl-CoA, and activate fatty acid synthesis and activated storage of that energy as triacylglycerols. Regulation of fat metabolism by glucagon and epinephrine. Remember, they tend to have the opposite effects as insulin. So glucagon and epinephrine result in the stimulation of triacylglycerol breakdown, also known the mobilization of fatty acids. So we want to mobilize fatty acids when we need energy and use them for energy. We want to store fatty acids when we have plenty of energy and can store them. So glucagon and epinephrine result in stimulation of triacylglycerol breakdown and also decreased glycolysis and increased gluconeogenesis. So it's going to increase gluconeogenesis in the liver, right? Because if you need sugar in your periphery or in your blood because you have low blood sugar, you're going to increase gluconeogenesis in the liver. Because remember, your liver and your kidneys are really the only two organs that can make the complete pathway of sugar. So decreases glycolysis also in the liver. So rather than burning the sugar in the liver, you're going to secrete that sugar into the blood. So triacylglycerol breakdown and the resynthesis, again, they create a futile cycle. 75% of free fatty acids released by lipolysis are reesterified to form triacylglycerols rather than be used for fuel. So we do this. Some recycling occurs in the adipose tissue, and some free fatty acids from adipose cells are transported to the liver, remade into triacylglycerol, and then redeposited in the adipose system. So it is a futile cycle that we have. Distribution between these two paths may vary. Overall, the percentage of free fatty acid being esterified remains at about 75%. So then we have this triacylglycerol cycle. Adipose tissue will have a triacylglycerol be broken down into a fatty acid, which can be then sent to the blood, used for fuel, or taken up by the liver, where that fatty acid will be remade into a triacylglycerol and delivered back to the adipose tissue, where lipoprotein lipase will strip off the fatty acids and then convert them back to triacylglycerols for storage. So we get this futile cycle that does occur. But we want to have fatty acids available for fuel if we need them, right? So recycling continues even in starvation. Benefits is that energy reserved for fight or flight crisis might occur during fasting. We still need to have energy to save our lives even if we're hungry, right? The total number of free fatty acids in flux may change, but the percent recycled remains unless a pharmacological intervention happens, such as thiazolidinone drugs. I'm bad at saying these words. So what is the source of glycerol 3-phosphate needed for fatty acid reesterification during lipolysis? Stimulated by glucagon or epinephrine, glycolysis is inhibited. So we have dihydroxyacetone phosphate is readily available to make that glycerol 3-phosphate. And the adipose cells don't have glycerol kinase to make glycerol 3-phosphate on site, so cells make dihydroxyacetone phosphate via glycerol neogenesis. So glycerol neogenesis contains some of the same steps as gluconeogenesis, converts pyruvate to dihydroxyacetone phosphate, and basically it's an abbreviated version of gluconeogenesis in the liver and the adipose tissue. Explains why adipose cells express pyruvate carboxylase and PEP carboxykinase, even though fat cells don't make glucose. So these are the, the glycolytic or the gluconeogenesis enzymes that overcome that pyruvate kinase reaction, or PEP kinase, or pyruvate kinase. Yeah. So glycerol neogenesis, we have pyruvate, gets converted to oxaloacetate because we add that carboxylic acid, and that activates the pyruvate to oxaloacetate. Phosphoenol pyruvate then is made from the oxaloacetate via PEP carboxykinase. And then that phosphoenol pyruvate ultimately will get converted to dihydroxyacetone phosphate. So that'll be glycerol 3-phosphate dehydrogenase will convert that to glycerol 3-phosphate, which can then be used for triacylglycerol synthesis. So the regulation of phosphoenol pyruvate carboxykinase expression is tissue dependent. Cortisol and glucagon both increase phosphoenol pyruvate carboxykinase expression. Results in more triacylglycerol synthesis, so more is released into the blood. So cortisol and glucagon. Do you guys know what cortisol is for? It's a hormone. It's one of your stress hormones, right? So that's why, they say, living a stressful life tends to cause you to build fat stores because it results more in triacylglycerol and you get more released in the blood. Cortisol and other glucocorticoids decrease phosphoenol pyruvate carboxykinase expression in the adipose tissue, which decreases glycerol neogenesis in the adipose, means less recycling. More free fatty acids are released into the blood. Most glycerol freed from triacylglycerol in the adipose is sent to the liver and converted back to glucose. Cortisol and glucagon can elevate blood sugar. So increased phosphoenol pyruvate carboxykinase, or should I just say PEPCK? See, I can't even say that. Expression in the liver increases gluconeogenesis, so you have increased glucose production in the liver too. Decreased PEPCK expression in the adipose tissue yields more glycerol freed, sent to the liver, converted to glucose, so it boosts your blood glucose levels. Plus the free fatty acid associated with the influx through the triacylglycerol cycle interferes with glucose uptake in the muscle to keep glucose, blood glucose high. Thiazolidine drugs target insulin resistance by increasing glycerol neogenesis. Elevate free fatty acid levels seem to promote insulin resistance. Thiazolidinones or glitazones upregulate PEP carboxykinase in the adipose tissue, which leads to increased glycerol neogenesis, increased resynthesis of triacylglycerols in the adipose tissue, which decreases the release of free fatty acids into the blood. Thus, the drugs promote sensitivity to insulin because there's the glycerol that was in the adipose isn't being kicked out, going to the liver, being made into more sugar to be kicked out into the blood, and the free fatty acids aren't interfering with the glucose uptake by the muscle. And this is the structure of those thiazolidinones, or the glitazones. And this shows you the regulation of glycerol, or the regulation of glyceroneogenesis via glucocorticoid hormones. And then how the thiazolidinones interfere with that. Chapter's huge. So attachment of the head group to the diacylglycerols, we're going to need lipids for membranes. We have a diacylglycerol. If you attach a phosphate, it makes phosphatidic acid, and then we're going to attach an alcohol to this, and that will give you some head group. And there's your glycerophospholipid. Further details on attaching the head group. So either one of the alcohols is activated by attaching a cytidine diphosphate. The free, not bound to the cytidine diphosphate alcohol is then does nucleophilic attack on the CDP activated phosphate and releases CMP and a glycerophospholipid. So here we have the activated diacylglycerol phosphatidic acid with a CDP added. CDP diacylglycerol. You have a head group that has an alcohol group, attacks this phosphate, releasing CMP, and then you have glycerophospholipid with a head group. Or strategy 2, you can have the alcohol from the diacylglycerol attack the head group that's already bound to the phosphate of the CDP, releasing CMP. So CDP diacylglycerol is a precursor to phosphatidylserine and derivatives in E. coli. CDP diacylglycerol is attacked by the nucleophilic hydroxyl of serine, which then yields phosphatidylserine. So you have the alcohol group of the serine attack the CDP, and that connects the serine with the diacylglycerol, and you have phosphatidylserine as a result. And this is catalyzed by phosphatidylserine or PS synthase. Phosphatidylserine can be decarboxylated because it's got a carboxyl group on the other end of that serine and an amino group. And that, when you decarboxylate that carboxyl group, because remember serine's an amino acid, that forms phosphatidylethynolamine, and that's catalyzed by phosphatidylserine decarboxylase. The CDP diacylglycerol's activated phosphate is then attacked by glycerol 3-phosphate, phosphatidylglycerol 3-phosphate, catalyzed PG3-phosphate synthase. PG3-phosphate is hydrolyzed, and that yields P-glycerol. Catalyzed by PG3 phosphate phosphatase. Phosphatidylglycerol dimerizes to form cardiolipin, catalyzed by cardiolipin synthase. And here you have the dimerization. Here you have cardiolipin then, catalyzed by cardiolipin synthase. And this is how you form the phospholipids in E. coli. So you can form synthesis of anionic phospholipids in eukaryotes, similar strategies to that used in E. coli. Phosphatidylenidazole is made from CDP diacylglycerol and enidazole. And you have a specific kinase that makes phosphorylated derivatives. The difference from bacteria is cardiolipin is made from attack of phosphatidylglycerol on the CDP diacylglycerol rather than from 2 phosphatidylglycerols. So let's see what we have here. This is cardiolipin, this is phosphatidylenidazole. For cardiolipin synthase, we have the phosphatidylglycerol that comes in and attacks to form this kind of like dimer of 2 diacylglycerols, right? And phosphatidyllenidazole comes in and does the attack, and then you have phosphatidyllenidazole. You guys remember what this is important for? It's the signaling lipid involved with G protein coupled signaling. This gets cleaved by phospholipase C, releasing IP3, where you get these are esterified with phosphate, and you have inositol triphosphate acts as an intracellular, secondary messenger.